the contamination of the food crops here discussed. Almost 25% of the total contaminated crops are contaminated due to mycotoxins. And due to veterinary drugs, there are 9% agricultural crops are contaminated. And 30% of the contaminated crops are due to heavy metals. Now, why we deal mycotoxins? The origin and the discovery of mycotoxin is in 1960. The discovery of mycotoxin as well as the discovery of most carcinogenic mycotoxins, aflatoxins. According to the World Health Organization and Food Agriculture Organization of the United Nations, 25% of the world crops contaminated due to mycotoxins. And in almost 77 countries, there are regulatory limits for the mycotoxins as well as for the aflatoxins. And in 13 countries, there is no regulatory limit for the mycotoxins. Today, almost 400 different types of mycotoxins discovered, in which 150 species of fungi and new ones are discovered. So, what is actual mycotoxin? Myco mean of fungal origin and the toxin, as you know, poison. Mycotoxins actually basically are the secondary metabolites produced by the molds and the fungus, and contaminate the agricultural products that are the most we use in the human food or in the animal food, and are the cereals that are the staple food for the human being. And what is the condition for the production of mycotoxins? Different tropical conditions are compulsory for the production of mycotoxins, such as high temperature, high humidity, and high moisture content, and flashy rains and unseasonal rains are causes for the production of mycotoxins. Here comes a series scenario in Pakistan. During there is a very dominant separation of the production and the crop area and the production area and the production and the area of harvesting in different years. Here we see that production of the agricultural products is varied due to the mycotoxin contamination. Now, the general presentation for the mycotoxin presentation is as follows. Here, from the spore of the fungus and the molds are accumulated during construction, but remain inactive many years by tropical conditions such as humidity and the temperatures. These spores and these molds are activated. Here is the brief presentation of the mycotoxins. Now, these mold and mycotoxins are contribute a layer on the surface of the household buildings, and when contact with the skin make irritation, and if inhaled and accumulate in the lungs. And if we further discuss on mycotoxin, it may bind with the DNA and cause short brain and may disturb the signal brain and neurosystem of the human. Here, mycotoxin bind with the DNA and then in the blood tissues and travel in the streams of the blood and cause disruption of the brain signals. Now, what we need? A good exercise of the and testing is needed for mycotoxin control during construction. So, common family members of the fungal mycotoxins are aflatoxin, ochratoxin, patholine, trichothene, fumalism, and alternaria. And today we discuss the most important class of mycotoxins as aflatoxin, the number one carcinogenic class of mycotoxins. So, mycotoxins are present in all stages of food, during processing, during production, and during storage of the food. The natural toxins occur primarily in these cereals, which are the main staple food of the human being or are the main staple food for the animal kingdom. How was the concept of mycotoxin developed? In 1891, there was a report of moldy rice in Japan, and next in 1960, there is a turkey pox disease, and almost 10 million poultry birds died in this disease. So the investigation is what was find the trace of the mycotoxin, and today we know this mycotoxin as aflatoxin, number one carcinogenic class of mycotoxins. Here is the cyclic process of the mycotoxins, where the mycotoxins are entered in food chain. Infection of plants by mold is directly linked contaminated the food of plant origin, and in other way, infection of plants by molds then contaminated the feed of the animals, and this contamination is further go into the toxin into the blood and organs, and it ultimately contaminated the food of animal origin. And then there is from the animal another aflatoxin that is called aflatoxin M1, that is metabolized form of aflatoxin B1 is entered into the human body. Now the aflatoxin. Aflatoxin is produced by a fungus called Aspergillus flavus and Aspergillus pernicious. Aflatoxin is a white crystalline solid that is optically active and classified as a carcinogenic group. Now the favorable conditions and factor for the mycotoxin severity. Harvesting of over-matured crops is caused by the mycotoxin and caused by the aflatoxin. Mechanical damage of pods of the and high temperature. Humidity greater than the 80% is also cause for the aflatoxin production. Stacking harvest during high humidity, bad weathering conditions such as unseasonal rains and monsoon season also causes for the aflatoxin production. High moisture content of cereal and effect of high seed constituent is also responsible for the aflatoxin production. And contamination limits and occurrence. There are different regulatory limits for the occurrence and the contamination limits for the aflatoxins. Allowable limits in rice and cereals, different cereals, wheat, corn, and wheat is 0.5 parts per billion. And according to the European Union, there is a limit in 1998, 0.2 parts per billion for foodstuff. And aflatoxin B1, occurrence of aflatoxin B1 reported in Pakistan in chilies is 1 to 79 parts per billion, and aflatoxin reported in India in corn is 5 to 666 parts per billion. There is occurrence of effects of aflatoxin on crops. The first is main concern we deteriorate quality of the crops, and the second one is contaminated food, animal food or human being food. And the third one is the effect of international trade, which importance of the international trade. Now the chemistry of aflatoxin. There are major 4 types of aflatoxin classified as aflatoxin B1, aflatoxin B2, aflatoxin G1, and aflatoxin G2. These classification is because aflatoxin is showing blue color, and that is known as aflatoxin B1 under UV light. Aflatoxin B1 and B2, and showing a green color, so we call it aflatoxin G1 and G2. And the aflatoxin B1 is further metabolized and entered into the milk, and we call it aflatoxin M1 and M2. Now there are the different aflatoxin structures for aflatoxin B1, B2, aflatoxin G1 and G2. Now the mode of action for aflatoxin. There are 3 possibilities for the mode of action of aflatoxin. These are mutagenic, carcinogenic, and teratogenic. And there are 2 types of diseases called acute diseases and chronic diseases for the mode of action and mechanism of aflatoxin. In first case, if the aflatoxin is bind with the DNA, then it is called a chronic disease. If it hydrolyzed, now if the aflatoxin is hydrolyzed and goes into the milk and aflatoxin B1, then it is called aflatoxin M1. And if the aflatoxin epoxide is formed at the 8-9 position, then it binds with the DNA and causes few diseases. Now there is another possibility for the mechanism. This is the aflatoxin epoxide is further metabolized and hydration is take place. Then aflatoxin dihydroxy aflatoxin derivatives formed, and this goes to the interaction with enzyme lysine residue, and this causes acute toxicity of the aflatoxin. Now the effect of aflatoxin on health: it binds with the DNA, and this causes problem for DNA replication, and mutation in genes that control the life cycle can lead to the cancer. Now the effects of aflatoxin on health. Adverse effect of aflatoxin on human health and livestock, international trade, and the blood tests have been showed very high percentage of the population of several countries in Asia and Africa are exposed to aflatoxins. Now we discuss another disease, aflatoxicosis. That is primarily hepatitis disease, which is mainly concerned with the aflatoxin. Now the mycotoxin severity conditions. There are type of mycotoxins and exposure duration and dose, which may suggest that acute diseases or the chronic diseases of the mycotoxins. Age limit, nutritional status and health of individual, and the synergetic effect with other chemicals or mycotoxin, primary target organs, liver, lungs, kidney, nervous system and immune system. Now the control. There are different physical and chemical methods for the prevention and for the minimization of mycotoxins and aflatoxins. Here are good agriculture practices. Minimize the production of mycotoxins. Drying techniques also minimize the production of mycotoxins, and proper storage facilities of the agricultural commodities are also minimize the production of mycotoxins, and caring during transport and the marketing of the agricultural commodities are also control the and minimize the production of mycotoxins. Now the method of analysis. Sir, last slide. And there are three methods. The TLC method is the using for the analysis of the aflatoxins. This method is used for the cost concentrate and using silica gel plates. Now the most increasingly and acceptable method for the detection and analysis of the mycotoxin is TLC, thin layer chromatography with high pressure liquid chromatography, and is another method. ELISA method is also used for the analysis and for the analysis of mycotoxins. And reference. Thank you.